Hi, my name is Professor David Jones and I'm from Cardiff University School of Biosciences, and today I'm going to talk about a protein called green fluorescent protein, which is one of the most important proteins currently in use in the biosciences. Now, green fluorescent protein is comprised of about 238 amino acid residues and exhibits bright green fluorescence when exposed to blue light. In other words, when blue light is shone upon on this protein, it emits a green light. Although many other marine organisms have similar green fluorescent proteins, GFP traditionally refers to the protein first isolated from the jellyfish Aquaria victoria, as you can see here on the screen. The jellyfish itself lives in the ocean, so as a result, not much light gets down to where the jellyfish normally lives. So how can the jellyfish emit light if there's no incoming light for it to use? Well, the way it gets about this is by a process called chemiluminescence. In this case, light is emitted due to a chemical reaction. And in this case, that occurs by its partner protein, aquarin, which interacts with metal ions, which induces a blue glow. Now, some of this luminescent energy is transferred to GFP, shifting the overall colour towards the green. So GFP uses that light to excite itself. However, its utility as a tool for molecular biologists did not begin to be realised until 1992, when the clone in a nucleotide sequence of GFP was published. The coding sequence of GFP was first expressed in a bacterial cell called E. coli and in a simple worm organism called C. elegans. Remarkably, GFP molecule folded and was fluorescent at room temperature without the need for exogenous cofactors specific to the jellyfish. This suggested it was a naturally fluorescent protein and needed no cofactors or substrate in order for it to work. And this was quite crucial because it meant you could move that gene outside the jellyfish and put it into any organism you wanted. And we could do that by using the gene sequence, because we can easily transfer genes from one organism to another. And the key thing is then a functional protein was produced. Although this GFP was fluorescent, it did have several drawbacks. It was not ideal as a fluorescent protein. It wasn't that bright compared to a lot of other chemical probes that were around at the time, and it had a lot of sensitivity issues to various things. And it had problems actually working at 37 degrees. So these factors subsequently have been addressed by protein engineering, and GFP now is one of the most widely used tools in cell biology. And the importance of GFP meant in 2008, the Nobel Prize in chemistry was awarded to these three individuals. Shimamura was the first person in the 60s and 70s who looked at aquarin and GFP as the basic sort of fluorescent units. Roger Shen basically took the gene sequence and made a lot of useful fluorescent proteins out from it. Martin Schäffli was one of the first people to show it could be used in a different organism. And one of the sad stories is the person who actually cloned the gene, Doug Prasher, he ended up having to leave science because nobody would fund him for his work, and he ended up being a courtesy driver for a car firm, a car sales firm in the US. So not everything ends in a fairy tale ending in these situations. So if we look at what GFP is useful for, and what it's been mostly used for, is as a genetically encoded tag for cell imaging. What that means is we can take the GFP gene and genetically splice it onto the end of another gene, because most proteins we can't actually see inside the cell. So we need to make them visible somehow. And that's where GFP comes in. It's what we call a fusion product. And what we normally do is put GFP at the end of another protein, and it acts as sort of like a molecular tag. And you can, because you can see where GFP is, because it's fluorescent, you can follow that other protein around. The thing is though, GFP is another protein. So as a result of that, it does have its own particular bulk associated with it. And I think a lot of people don't realise that when you have a protein fused to GFP or any other fluorescent protein, it is carrying that around, and it can slightly impact the function of that other protein if it's not designed properly, or that fusion is not designed properly. So I do suggest to people who create GFP fusions is to give a little thought about how that fusion is created and not just whack it on the ends of the protein. But the great thing about it is it does then show up individual proteins quite clearly inside the cell using a process called fluorescence microscopy. And this is, we have a bioimaging hub here at Cardiff, and these are some of the images they take with GFP labeled cells, and you can see the greenness here. That's what the proteins are labeled with GFP, so only particular proteins are labeled. But we also can use it in combination with other dyes, such as one that stains the nucleus. So we can see how different compartments in the cell, but if we looked up by eye, would just look amorphous. So it's a very important aspect for how we investigate biological systems. And the key thing is this genetic tagging here. So it's very, very good for in vivo labeling of proteins. It's what we call a good spatial marker. It allows us to look at where things are inside the cell and look for how proteins move during the course of their lifetime. It also acts as a good temporal marker, and that is, how do genes change expression over time, and therefore protein levels over time. So it gives us an idea of when a gene is switched on or when a gene is switched off, or when levels of protein change flux over time. So there we go, that's a basic introduction to green fluorescent protein.